Since we have discussed the sensory system and discussed the general senses, let us also discuss the special senses, particularly the important ones like vision, hearing, equilibrium. Starting with the visual sense. There are three layers of the eyeball. The sclera, choroid, and retina. The sclera in front forms the transparent structure. Most anteriorly, it has the transparent structure of the outer coat, the cornea. Choroid, the middle coat or the middle layer, it has the pigment. In front, anteriorly, it forms a curtain-like iris, and at the center of the iris, there is an aperture, the pupillary aperture. Iris then is also associated with the ciliary body and ciliary muscle. The ciliary body and ciliary muscle have the suspensory ligament which suspend the crystalline lens of the eyeball, the crystalline lens of the eye. That's the structure for the middle coat, and then the inner coat would be retina. Light rays coming from an object will make an inverted image on the retina. There are neural elements in the retina which the image formed will be converted into electrical impulses which will then be carried by the optic nerve to the visual cortex in the occipital lobe. That's in summary the structure of the eyeball. There are two media as well. The aqueous humor in front of the lens, the aqueous humor, and behind the lens, the vitreous, jelly-like vitreous humor or vitreous body. Let us see the optics of vision. There are four surfaces where refraction occurs. Refraction is bending of the light rays. When it traveling from one medium, they enter another medium. There is bending, which is the refraction. So, in case of our eyeball, refraction occurs at four different surfaces. Anterior surface of the cornea, where light rays are entering from air to cornea. Then posterior surface of the cornea, because light rays will be entering from cornea to the aqueous humor. And then anterior surface of the lens, because light rays entering from aqueous humor to the lens. And then posterior surface of the lens, the light rays exit the lens and enter the vitreous, and finally form the image on the retina, the inverted image. Now, this is a complicated model of the eye. First of all, out of all these, the greatest refraction occurs at the anterior surface of the cornea. The maximum refraction will occur at the anterior surface of the cornea. Reason being that refraction depends on the difference in the refractive indices of the two media, and the maximum difference is between the air and the cornea. The difference in the refractive indices is maximum between air and the cornea. So greatest refraction will occur at the anterior surface of the cornea. Now, since this is a very complex model of the eye to understand, therefore a simplistic model of the eye was devised. This simplistic model of the eye, called as schematic eye or reduced eye of Listing. Reduced eye or reduced eye of Listing. It's a simplistic design of the eye. Now, in this, all four planes where refraction occurs, are algebraically added to consider only one single plane of refraction. This plane is said to be about 1.7 millimeters behind the cornea in the aqueous humor, and only single refraction will be taking place according to this schematic model of the eye. And the entire refractive power of the eye would be about 61 diopters. There is a range 59 to 61 diopters, out of which 41 diopters is by the cornea and about 18 to 20 diopters is the dioptric strength of the lens. So reduced eye of Listing has these descriptions, that single plane of refraction is considered instead of 4, adding all 4 algebraically, and its power would be well, there is a variation in this figure from plus 59 to up to plus 61 diopters. So out of that, plus 41 diopters is at the cornea, and the dioptric power of the lens, the crystalline lens, is plus 18 to 20 diopters. So that's how it is, plus 59 to plus 61 diopters. Now, image formation has various problems, and these are called as errors of refraction. So errors of refraction: myopia, the short-sightedness when image is formed in front of the retina, because of the commonest reason being that the anterior-posterior curvature of the eyeball is increased. So image is formed in front of the retina. Treatment is placing the biconcave lens so that there would be divergence of the light rays as they enter the eye, and then as they converge, image will be formed on the retina. Hypermetropia. Hypermetropia would be long-sightedness or far-sightedness when the person can see far-off objects clearly but cannot see the nearby objects clearly. Now, let us just define what are the near objects and far objects. 6 meters is said to be the boundary. The objects which are within 6 meters from the eye are considered as near objects, and beyond 6 meters up to infinity is called as the far object. Why 6 meters is the boundary? Is because 6 meters and beyond, if the object is 6 meters or beyond, that then the parallel rays coming from such an object will automatically converge on the retina without having to do any changes in the eye. That's therefore 6 meters and beyond up to infinity, that will be the far distance, far object. And if the object starts coming closer within 6 meters, then some changes in the eye become necessary in order to have image on the retina. Those changes are called as accommodation reaction. The changes of accommodation or accommodation reaction, also called as accommodation convergence reaction. So, as the object starts coming closer and closer to the eye, what happens is the image starts forming beyond the retina, and therefore, in order to form the image on the retina, the curvature of the lens and dioptric power of the lens needs to be increased. This is achieved by the contraction of the ciliary body and ciliary muscle, so that the suspensory ligaments, they release their tension on the lens and the lens curvature will be increased. So lens power is increased. three changes that accommodation, these changes in order to see the nearby objects clearly is called as the changes of accommodation. And there are three changes that occur in this accommodation reaction. First is increased power of the lens, so that the convergence of rays is much greater and the image forms on the retina. Second is medial rotation of the eyes. There is medial rotation of the eyes, convergence of the eyes, so that light rays coming from the center of the object, from the object, they pass through the center of both pupils. So eyes rotate medially. And third is constriction of the pupil, so that peripheral light rays are cut off and only the central rays coming from the object, they pass through the pupil. So these are the three changes that occur in the accommodation. Accommodation reaction, it involves cortex, so generally it's not called accommodation reflex but reaction. That as you look at the near object, initially since image is not formed in the retina, a blurred image formed, signals are sent to the cortex, and from the cortex, due to the blurred image, the signals are sent back to the oculomotor nerve, the third nerve, Edinger-Westphal nucleus, and then a third nerve nucleus would bring about those changes of accommodation. So that's accommodation, to see the near objects. Now what is near point of vision? Near point is that closest point from the eye at which the object can be seen clearly. I mean, if the object comes closer than that, then again the image will be blurred. So that closest point from the eye at which the object placed can be seen clearly. Object cannot come closer than that. If it comes, the image will not be clear again, it will be blurred. So why is that near point? Because at that point, the closest point from the eye, there is maximum contraction of the ciliary body. There is a greatest contraction of the ciliary muscle, and therefore that is the greatest curvature which is achieved for the lens. I mean, there cannot be any further increase in the dioptric power of the lens, and therefore that's the maximum. Therefore that is the closest that the object can be seen clearly. So near point is that point of the vision, and amplitude of accommodation is said to be the distance between far point and the near point. Just one additional factor, when a child is born, generally they are born with axial hypermetropia. Coming to the near point and accommodation reaction, there is something called as Argyll-Robertson pupil. Argyll-Robertson pupil, quite often seen in neurosyphilis or such conditions. And how this pupil is described is accommodation reaction is present, but pupillary reflex is absent. Accommodation reaction is present, but pupillary light reflex is absent. Now, pupillary light reflex, as we will see little later, requires the tectum and pretectal nuclei. So, possibly the lesion is in the pretectal nuclei, and therefore pupillary light reflex is absent in such a pupil. Now, hypermetropia, image is formed beyond the retina, and therefore it is corrected by placing the convex lens in front of the eye so that there is convergence of the light rays, and then they further converge as they enter the eye and image will be formed on the retina. This normally occurs because of the increased vertical diameter of the eyeball. So retina slightly has come forward, or therefore image being formed beyond the retina. Presbyopia, well, there is an easy way of remembering. Very often students tend to confuse this. Myopia is short-sightedness, means what you can see or what you cannot see. So simple way of remembering is myopia is mypia, means something which is closer and you can see the closer objects clearly but cannot see the far off objects clearly. Hypermetropia is hypia, somebody you are waving at a distance, so hypermetropia, so long-sightedness, far off objects and can be seen clearly. Then presbyopia is probably bye-byepia, bye-bye to the power of accommodation after the age of 40. So what's presbyopia? Presbyopia is generally the defect that occurs due to the aging process. After the age of 40, there is loss of power of accommodation. Because of there is hardening of the lens protein coagulates and hardening of the lens, therefore lens curvature cannot increase beyond a certain point. Therefore power of accommodation is lost or decreased rather, and that means near point recedes from the eye, near point goes away from the eye, near point recedes from the eye. So that's presbyopia. The treatment is that bifocal lens is used for the far vision and for the near vision. And then astigmatism, in astigmatism there is irregular shape of the cornea or lens. Cornea or lens may become egg-shaped. Egg-shaped cornea or lens means what? It has two surfaces. You see, a normal curvature of the cornea would be like this. So light rays, their refraction would be with the same angle, whereas egg-shaped cornea has two planes of surfaces, and therefore refraction is not with equal angle, and therefore the light rays do not meet and do not create a clear-cut image on the retina. The treatment of this would be correction in one plane by spherical lens and correction in the remaining plane by the cylindrical lens. How do we check the vision? Distant vision, near vision. Near vision is tested clinically by the Jaeger's chart, and distant vision is by the Snellen's chart. So before we talk about the Snellen's chart, let us try to understand the visual acuity. What is a visual acuity? Visual acuity is described in terms of minimum separable. Minimum separable means 2 points should be separated by such a minimum distance that they can be seen separately. If they come closer than that, then those two objects will not be seen clearly. Their images will be fused. And that means one won't be able to see the details of an object. After all, why do you have to see any 2 points separately? In order to understand the details of that object clearly. So, visual acuity is expressed in terms of minimum separable distance or minimum angle subtended by the 2 points. So here are the 2 points. We can see them separately only when they subtend an angle of 1 minute or more than 1 minute. This is just diagrammatic. Obviously, this is not 1 minute. This is just a diagrammatic presentation for the sake of understanding. If the two objects subtend an angle of 1 minute, means they are separated by such a distance that they subtend an angle of 1 minute or more than 1 minute, then those two objects can be seen clearly. So a minimum of this much distance is necessary. They have to subtend this 1 minute angle at the nodal point of the eye. Nodal point of the eye is said to be at the junction of anterior 2/3 and posterior 1/3 of the lens. So now nodal point, these two objects should make an angle of 1 minute or more. What is the meaning of this 1 minute angle? 1 minute angle, now that's important to understand. 1 minute angle at the nodal point of the eye, it corresponds with roughly 4.5 micron distance on the retina. So these two objects, if they subtend 1 minute angle, this is just diagrammatic, then they are separated by such a distance that their images on the retina will be separated by 4.5 micron distance. Now, what is the meaning of this 4.5 micron distance? You see, width of a single cone is about 2.5 to 2.8 microns. That's the width of a single photoreceptor. You know, photoreceptors in the retina, rods and cones, and this is the width of a single photoreceptor on an average. So when two images are separated by 4.5 micron distance on the retina, that means they are falling on two separate photoreceptors with at least one receptor left unstimulated between the two images. You can see here, let's assume that this is 4.5 micron distance, whereas width of a single cone is about 2.5 microns. So it ensures that at least one cone or one photoreceptor is left unstimulated between the two images. Only then you can see any 2 points clearly. So that's the meaning of visual acuity. I mean, imagine that the two objects come closer than this, then their images will also come closer. If they subtend less than 1 minute angle, their images will also come closer. They will fall on the adjacent photoreceptors, those images, and then those images will get fused. We won't be able to separate those 2 points. We won't be able to see those 2 points separately. So what is needed for visual acuity is differential excitation of the cones, differential excitation of the rods and cones or photoreceptors, that two cones stimulated and one cone left unstimulated between the two cones. That's the differential excitation of the photoreceptors. Now, this visual acuity is maximum at the fovea centralis. You see, near the central part of the retina, there is a depression called as macula lutea, and the central part of it, the fovea centralis. The visual acuity is greatest at the fovea centralis, best at the fovea centralis, and therefore when we have to examine the details of an object, we turn our eyes to that object. Why? Because we are looking at that object so that the light rays coming from that object will fall on the fovea centralis, where the vision is most detailed and accurate. Visual acuity is greatest. Why? There are a few reasons to it. So just try to understand the reasons. Fovea centralis has only cones, no rods, first of all. Those cones are slenderer, thinner, and densely packed. The cones are slenderer and thinner and densely packed. So imagine if the cone is slender, our prerequisite is that at least one photoreceptor should be left unstimulated between the two images on the retina. Only then we see the two separately. So if the cone is thinner, that means the two images can come closer because they have to leave less distance between them because the cone is thinner. Less distance will have to be left, and plus they are densely packed. So the two images can come closer. That means two objects which we are looking at can be closer, and we can still see them separately. So that's the importance of a thinner photoreceptor and densely packed photoreceptor. And the third reason is why fovea centralis has more detailed and accurate vision is cone to ganglion cell ratio, optic nerve fiber ratio, cone to ganglion cell ratio is 1:1 in the fovea centralis. One cone is synapses with one optic nerve fiber. So these are the reasons why visual acuity is most detailed in the fovea centralis. On the other hand, as you move towards the peripheral parts of the retina, two things happen. Peripheral parts of the retina, the population of rods goes on increasing. In the fovea centralis, there are only cones. Cones are designed for bright vision, rods are meant for dim vision. As you move towards the peripheral parts of the retina, population of rods increases. Rod is meant for dim vision. Secondly, many rods converge on one optic nerve fiber. Remember, in the fovea, the photoreceptor to optic nerve fiber ratio was 1:1. But as you go to the peripheral parts, many photoreceptors, almost 60 rods will converge on one optic nerve fiber, which means what? Their receptor potentials will be summated. So the point is, near the peripheral parts of the retina, the visual threshold is low. Visual threshold is low. The visual acuity is less there in the peripheral parts, but visual threshold is low means by a lower amount of light, the peripheral parts are likely to be excited as compared to the central part of retina. Peripheral retina will be excited with a lesser amount of light. It can be detected much earlier or much better in the peripheral parts of retina. The reason is 60 rods converging on one optic nerve fibers as you move towards the periphery of the retina, and their receptor potentials get summated with a few photons of light also, and that will therefore even less amount of light will be detectable in the peripheral retina. That's about the visual acuity. It is tested by Snellen's chart. Snellen's chart. Now, one question that is never asked and answered is, if you look at the Snellen's chart, Snellen's chart is placed at 6 meters distance because 6 meters is our far point of vision. From 6 meters up to infinity is the far point of vision. Fine. Snellen's chart is therefore placed at 6 meters, and the lowermost line is to be read from a 6 meter distance. Why the lines above, except barring 9 meters, the above lines are 12, 18, 24, 36 and 60 meters. The letters are constructed in such a way that those letters, for instance, 12-meter distance is written. That means those letters should be read from a distance of 12 meters. 18 meters line means those letters should be read clearly by a normal vision person from 18 meters distance. Why these distances are, why the letters have been constructed in the distances of multiples of 6, barring this 9 meters. Why so? The reason is, the normal person can read the lowermost, the last line from 6 meters. So if the patient who is reading it is also reading it from 6 meters, you write the vision as 6 by 6. Now listen to this. If the subject is reading only up to the 18-meter line and nothing beyond, nothing below that, then you write his or her vision as 6 by 18. What is the meaning of this? The meaning of this is, this person's minimum visual angle has become reciprocal of the Snellen's notation. If this person's vision is 6 by 18, that means now his minimum visual angle is reciprocal of the 6 upon 18. That is 18 upon 6, that is 3 minutes. So for a normal person, the minimum separable is defined as two objects subtending an angle of at least 1 minute. Then they can be seen separately. This person, two objects will be seen clearly when they subtend a 3-minute angle or more. The minimum angle should be 3 minutes. That means his visual acuity has been reduced. So to check this angle, to calculate this angle, determine this minimum angle, the letters have been constructed in such a way in the multiples of 6 meters distance. A person whose visual acuity is 6 by 60, for example, that means his minimum visual angle will be 60 upon 6, and that is 10 minutes. So if this person will be able to see 2 points separately if they subtend an angle of 10 minutes or more at the nodal point of the eye. That's the Snellen's chart, and how we interpret the Snellen's chart or Snellen's notation. Let us see the color vision, some of the important aspects of color vision. There is a theory called as Young and Helmholtz theory. Trichromatic theory for color vision. Most accepted theory is Young and Helmholtz trichromatic theory for color vision. According to this theory, there are 3 cones, red, green and blue, and those colors, for example, red monochromatic light, it will excite the red cones by 100% stimulus value, but green and blue cones will not be stimulated at all, and it will be perceived as a red light. So 3 cones, 3 different types of cones, and 3 basic primary colors, and that's how various other colors will be perceived by different stimulus values for this. For example, orange monochromatic light will stimulate red cones with a stimulus value of 99%, green cones with a stimulus value of 42%, and blue cones maybe a stimulus value of 0%. So 99, 42, 0, this stimulation of these 3 cones, this combination is perceived as the orange monochromatic light. So this is how we perceive light, the color, by stimulation of different cones differently. And then color blindness, these three cones, these three colors rather, are given numbers 1, 2 and 3. So the trichromats will have all the three colors, all the three cones normal. That will be a normal vision. Dichromats means they have two colors intact, and there is a color blindness for one color. So if it's a red color blindness, now red color as if given number 1, and if that that cone is absent genetically, if it is absent, then it will be called as protanopia. So three types: protanopia, deuteranopia and tritanopia. Number 1 color blindness, number 2 color blindness, number 3 color blindness is respectively called as protanopia, deuteranopia and tritanopia. These are dichromats, means they have two colors intact and only one color cone is missing. The monochromats have only one color intact and there is a defect or blindness for two colors. For example, monochromat, common example is red green color blindness. So it will have red and green cones absent, red green color blindness. This is called as a monochromat. Color vision is tested by the Ishihara's chart. Ishihara's plates or pseudo isochromatic plates, they will detect the color vision and color blindness or color weaknesses. One more aspect of our vision is transduction. How is it that the light rays, the electromagnetic energy, is converted into electrical action potentials in the optic nerve fibers? That is where you talk of photochemistry of vision. It's all about transduction of the light rays, electromagnetic energy converted into electric energy, action potentials in the optic nerve fiber. So basically a rod has these four segments: outer segment, inner segment, nucleus and synaptic body. Synaptic body synapses with the optic nerve fibers which are going to carry the electrical impulses. So it's like this: when light strikes the rod, there will be some reactions that occur in these outer and inner segments, and due to which finally an electrical current or electrical energy will be generated and will be transmitted by the optic nerve. What are those changes? Those are collectively called as photochemistry of vision. So rods contain the photosensitive pigment called as rhodopsin, which is a combination of opsin plus 11-cis-retinal. Now, when the light strikes the rod, in the outer segment of rod there are these discs or shelves where this rhodopsin is present, the photosensitive pigment. Now when light strikes, this 11-cis-retinal is going to be converted to all-trans-retinal, and then opsin and all-trans-retinal will start pulling away from each other. But before that, rhodopsin will go through successive stages of bleaching of rhodopsin. So it will be converted to batho-rhodopsin, lumirhodopsin, meta-rhodopsin 1 and meta-rhodopsin 2. Meta-rhodopsin 2 is called as activated rhodopsin. So batho-rhodopsin, lumirhodopsin, meta-rhodopsin 1, meta-rhodopsin 2. This is activated rhodopsin. After this, the opsin will start pulling away from all trans retinal. Now, once the all trans retinal has started pulling away from opsin, this gives rise to another enzyme cascade reaction, and that enzyme cascade reaction in the rod will cause finally, which by the way includes the protein transducin, will finally cause the sodium channels in the outer segment to be blocked. You see, normally in the darkness there is a sodium current or sodium cycle. Sodium is pumped out from the inner segment and it will go in from the outer segment through the channel. This is going on in the dark. When light strikes the rod and rhodopsin is decomposed or bleached, and it goes through the successive stages and opsin and all trans retinal is formed, then it gives rise to another cascade reaction, and finally due to that reaction the sodium channel will be blocked, so that now sodium is pumped out by the sodium potassium pump, but it cannot come back in. This will cause hyperpolarization of the rod, and that will decrease the neurotransmitter release, glutamate release at the synaptic body, and that is taken as a sense of light. We have already discussed this, that rod receptor potential is a hyperpolarizing potential. When the light strikes, rod is hyperpolarized, and that is taken as a sense of light. So that's the photochemistry of vision. Well, all trans retinal is also converted into all trans retinol, which is a storage form of vitamin A. All trans retinal getting converted to all trans retinol, which is a storage form of vitamin A. And then when the light is not falling on the retina, on the rods, all trans retinal will be converted to 11-cis retinal. Opsin will combine with it to form rhodopsin. Now this is when light is not falling, and in the complete darkness we have one more alternate route to resynthesize the rhodopsin, this all trans retinol. When not needed, when light was not falling, all trans retinal was converted and stored in this vitamin A storage form or all trans retinol. So when there is very less amount of light, and when you need extra rhodopsin because there are very less light rays available, then this will alternate route also will be made available. All trans retinol to 11-cis retinol. 11-cis retinol will be converted to 11-cis retinal, and then it will combine with opsin to form rhodopsin. So an extra pathway for resynthesis of rhodopsin. Why we need this extra rhodopsin? Because in the darkness there are very few photons of light, very less amount of light available. We need extra rhodopsin in that case. And that's the basis for night blindness. Vitamin A deficiency leads to night blindness because this extra storage form is not available, all trans retinol. So extra rhodopsin cannot be resynthesized. It cannot be synthesized in an extra fashion, so or in an alternative path, and therefore one cannot see clearly in the dark. That's the basis for night blindness. Let us just also talk about the dark adaptation, dark and light adaptation. What is dark adaptation? When we enter the darkness, initially we hardly can see anything, but slowly, as we have seen, this storage form of vitamin A or all trans retinol will provide an alternate route for resynthesis of rhodopsin. Extra rhodopsin will be available. So what will happen is retinal sensitivity to the light will increase because light rays are less. So the changes that occur in the dark are called as dark adaptation. What all changes happen? Pupillary dilatation. Pupil dilates so that since already there are less amount of light rays, pupil dilates to accommodate as many light rays as possible. Second is this: increased sensitivity of the retina to the amount of light. So this occurs within 25 to 30 minutes. Within 25 to 30 minutes, the sensitivity of the retina increases by 25,000 fold. Increase in retinal sensitivity is complete by 25 to 30 minutes after entering the dark. Retinal sensitivity increases by 25,000 fold. Initially, cones adapt. Cones adapt much faster as compared to the rods. Rods adapt much slower. This is called as the adaptation curve. Time in minutes. This is time in minutes, let's say by 25 to 30 minutes, and this is increased sensitivity of retina after entering the dark. So rods adapt slowly. That's the dark adaptation. Also, one question commonly asked is, why do the night vision goggles have red color, or why do the pilots use, while they are entering the dark, they are going to use red colored night vision goggles? That's because the red color provides a faster adaptation. The adaptation by red cones, and therefore for the red color, will be much faster, so that the pilots don't have to wait for the adaptation to take place very slowly, and it will remain for longer duration. The adaptability of the retina or increased sensitivity of the retina stays for longer duration. So red color goggles used as night vision goggles. Light adaptation is completed in 5 to 7 minutes. In about 5 to 7 minutes, light adaptation will be completed, and the opposite changes occur. There is constriction of the pupil and there is decreased sensitivity of the retina. That's the dark and light adaptation. The best adaptation in the light is for the yellow color. Fastest adaptation and longest adaptation is for the yellow color, and therefore you'll see life jackets are of yellow color. So remember, red color best for the dark adaptation, yellow color best for the light adaptation. In the light, as person enters the light, these changes are going to happen. Pupils will constrict. It will restrict the amount of light entering the eye, and sensitivity of the retina will be decreased. Rhodopsin will be bleached. So that's about the dark and light adaptation. Let us finally see the visual pathway, the structures associated with the visual pathway, and effects of lesions in the visual pathway. There is optic nerve, the second cranial nerve, which has two groups of fibers, the nasal fibers and the temporal fibers. Then there is optic chiasma. There is crossing over of the nasal fibers to the opposite side, and they are joined by the temporal fibers of the same side. So nasal fibers have crossed over to the opposite side. and they are joined by the temporal fibers of that same side. This is the optic chiasma, the crossing over of the fibers. The outer fibers, they have remained on the same side. Nasal fibers crossed over, and then that forms the optic tract. So optic tract is made up of nasal fibers coming from the opposite eye and temporal fibers of the same eye, same side. That's the optic tract. And then the optic tract, these neurons end in the thalamus. We have already seen this, that all the general and special senses have to pass through the thalamus. So lateral geniculate nucleus of the thalamus. And then from there, the next neuron arises and goes to the visual cortex in the occipital lobe. These are called as optic radiations. They are radially going fibers, optic radiation, also called as geniculocalcarine fibers, because from the lateral geniculate nucleus of the thalamus to the calcarine fissure in the occipital lobe. So it's a geniculocalcarine fibers or optic radiation. The visual cortex in the occipital lobe. Area 17 is the primary visual cortex, and 18 and 19 are said to be visual association areas. So 17, 18, 19, these are the areas in the visual cortex. Now, the structures associated with this main visual pathway. One is pretectal nucleus. Pretectal nucleus is involved in the pupillary light reflex. So, this visual pathway gives out a fiber that ends in the pretectal nucleus. The pretectal nucleus is connected with the Edinger-Westphal nucleus of the third cranial nerve. Edinger-Westphal nucleus of the third cranial nerve, and that's the pathway for pupillary light reflex. So when you throw the torch light in the eye, signals will be passed on by the visual pathway, and from there to the pretectal nucleus, and then to the Edinger-Westphal nucleus, the third cranial nerve nucleus. Then it sends the fibers back and causes constriction of the pupil. That's the pupillary light reflex, where pretectal nucleus is involved. Now, there is something called as direct light reflex. Direct pupillary light reflex means light thrown in one eye and pupil of the same eye constricts. And there is consensual light reflex. Throw the light in one eye and there is constriction seen in the other eye as well. Why is that? The reason is the pretectal nucleus is connected with Edinger-Westphal nucleus of the same side as well as of the opposite hemisphere, of the opposite side. So it will pass on the impulses to third nerve nuclei on both sides, and therefore throw the light in one eye and both Edinger-Westphal nuclei, I mean on both sides, they will be stimulated and there will be constriction seen in the other eye as well. That's one structure associated with the visual pathway. Another structure is suprachiasmatic nucleus of hypothalamus. Suprachiasmatic nucleus. Well, this nucleus of hypothalamus is concerned with circadian rhythm. So we will discuss more about this during the discussion on hypothalamus. But just to mention it here, visual pathway sends one set of fiber to the suprachiasmatic nucleus of hypothalamus, and from there the signals are sent to the pineal gland. So if it's a night time, we see it, the signals are passed on to the suprachiasmatic nucleus, and from there via superior cervical ganglion to the pineal gland, and then the melatonin secretion will increase in the night time or in the darkness. So this is concerned with our circadian rhythm. We will discuss about it more in the hypothalamus. These are some of the structures associated with the visual pathway. Finally, let us also see the lesions in the visual pathway. Effects of lesions. If there is a lesion on the retina, some lesion on the retina, somewhere in some part of the retina, then it will lead to what is called as scotoma. There will be a defect in the field of vision. Scotoma is defect in the field of vision. Well, there is a physiologic scotoma which is called as blind spot. You know, retina has this. I am talking about the normal vision. In normal people, this is other than the lesions that we are talking. In normal individuals, where optic nerve leaves the retina, there are no photoreceptors in that area where optic nerve is leaving the retina. So, light rays coming from a particular angle will fall on this part. And since there are no photoreceptors there, therefore, the light rays coming from that much area will not be visible. So, blind spot is that part, that area in the field of vision which is not visible when the gaze is fixed, because light rays coming from a particular angle are falling on the optic nerve head where there are no photoreceptors. So that's a blind spot, and blind spot is a physiologic scotoma, a physiologic defect in the field of vision. Scotomas are of two types, by the way. Negative scotoma means in that particular area nothing will be seen. Objects can be seen beyond that area and before that area, but not in that area. So that's a negative scotoma. Nothing can be seen in that particular area in the field of vision. Whereas positive scotoma means actually that much area shows black. It is completely dark. Nothing is seen, and in fact that area becomes dark. That's a positive scotoma. So if lesion on the retina, there will be scotoma. Then lesion of the optic nerve, lesion in the optic nerve. Then, as you can see, that there will be complete blindness on that side. Optic nerve itself is sectioned. So blindness of the corresponding eye. Of the corresponding eye. Then let us try to understand first the lesion transection in the optic tract. Now just see in the diagram that optic tract, let's say optic tract is sectioned over here. So what fibers are sectioned? Nasal fibers from the opposite eye and temporal fibers of the same eye. They have been sectioned. What were they carrying? See in the diagram. Nasal fibers, we all are aware that image is inverted on the retina. Why? Because the fibers coming from the nasal side of the eye, they fall on the temporal retina. Fibers coming from the temporal side of the eye, they fall on the nasal retina. Fibers coming from the upper part, upper field of vision, they fall on the lower retina. And fibers coming from the lower field of vision, they fall on the upper retina. That is how the image gets inverted completely. So you can see here that that these nasal fibers of the opposite eye, what were they carrying? They were carrying temporal field of vision of that eye. So temporal field of vision of that eye will be lost, and this lesion will cause loss of temporal fibers of the same eye. As you can see here, temporal fibers, what were they carrying? They were carrying nasal half of vision of this eye. This is the nose. So the person is not able to see half of the field of vision. In this particular case, as you can see, half of vision is lost for both eyes, but that half is for both eyes, it is on the same side. In this case, it's on the right side for both eyes. So it is called as hemianopia. Hemianopia is half vision is lost, and homonymous hemianopia, because both eyes are not able to see on the same side. In this case, if the person is standing like this, then half of the vision on right side is gone. So it's a homonymous hemianopia. Both eyes have lost the vision on the same side. Whereas if there is a lesion in the optic chiasma, let us imagine this lesion in the optic chiasma. Optic chiasma was crossing over of the nasal fibers, and temporal fibers remained on the same side. That was the optic chiasma. So if both nasal fibers are cut, what were they carrying? They were carrying temporal fields of vision of the respective eyes. See, the nasal fibers are cut. This is a nasal fiber that is cut. It was carrying both. They were carrying temporal fields of vision of both the eyes. Means then right eye will not be able to see on the right side, and left eye will not be able to see on the left side. Both eyes have lost their temporal fields of vision. So this is called as heteronymous hemianopia, because both eyes have lost half vision on different directions, or to be precise, bitemporal hemianopia. Both eyes have lost their temporal fields of vision. So these are the effects of lesions in the visual pathway. Let us now discuss the hearing and equilibrium, physiology of hearing and equilibrium. The organ for hearing situated in the inner ear. Let us first try to understand the anatomy for hearing. There is external ear, middle ear, three chains of ossicles, a chain of three ossicles, I beg your pardon, and then inner ear. This is the structure of the ear. Now, this is a diagrammatic presentation. External, middle, and inner ear has cochlea. Cochlea makes a 2.5 turn inward. This is a straightened cochlea. Actually, it is inward coiled by 2.5 turns. If it is straightened and straightened up like this, so this is the view of the cochlea which is straightened up. What are the functions of the middle ear? Two important functions, apart from, of course, conduction of sound from external ear to the inner ear. There are two important functions. One is impedance matching. Impedance matching. You see, the organs for hearing, receptors for hearing are in the inner ear, but they are bathed in a fluid in the cochlea. So sound waves have to first travel through the air, and then as they enter the inner ear, they have to move the fluids in the inner ear. They have to move through the fluids in the inner ear. This is a kind of impedance. It's a mismatch, impedance mismatch, and this is rectified by the middle ear. That middle ear mechanisms create 22 times more force 22 times greater pressure will be generated by the sound wave as it enters the inner ear. That sound wave pressure will be enhanced by 22 times. So that's increased enhancement by 22 times. That's called as impedance matching. And why it's needed? Because there are fluids in the cochlea. Sound will have to move through the fluids in the cochlea. So greater pressure will have to be generated if it has to pass through the fluids. How is this 22 times greater force is generated? See, there are three ossicles. The malleus, whose handle has been shown here in the tympanic membrane. Then incus and stapes. So, when the sound waves come through the external ear, there will be tympanic membrane vibrations, and these vibrations will be transmitted through the malleus, incus and stapes, and the stapes bone will move towards the oval window. These are the two windows of the inner ear, oval window and round window. So foot plate of stapes will move towards the oval window, and then sound will enter through this window, and it will enter the inner ear. So there are two factors that will generate, or rather increase, the pressure in the sound wave by 22 times. One factor is when the handle of malleus moves, the stapes moves with 1.3 times greater force. They are arranged in such a way that when sound comes and handle of malleus moves, the stapes moves with 1.3 times greater force. That is how they are leveraged. So that's one way of increasing the force, one factor. And second is, tympanic membrane has got a surface area of 55 millimeter square, and foot plate of stapes has got a surface area of about 3.2 millimeter square. So that means surface area of the tympanic membrane is 17 times the surface area of the foot plate of stapes. It means the vibrations of the tympanic membrane are going to be concentrated by 17 times as the sound waves enter the inner ear. So a concentration of 17 times, and therefore 17 into 1.3, that's 22 times. 22 times greater pressure is generated, or greater force is generated as the sound wave enters the inner ear. Another function of the middle ear is attenuation reflex. Attenuation reflex. Not all the sound waves need an increased pressure. Imagine a sudden loud sound burst, or a very loud sound with a high decibel intensity. Now what is likely to happen? Because of a too much pressure in that sound wave, imagine if that pressure is further enhanced, there is a possibility of damage to the cochlea. In such a scenario, the pressure in that sound wave needs to be dampened, and that dampening is called as attenuation reflex. The pressure in that sound wave is dampened to prevent the damage to the cochlea, called as attenuation reflex, occurs in loud sounds or sudden sounds. How is that achieved? First of all, attenuation reflex occurs within 40 milliseconds. It takes 40 milliseconds to develop. So what happens is, as soon as there is loud sound, there is contraction of two muscles in the middle ear. There is a tensor tympani muscle and there is a stapedius muscle. They contract, and because of that, the handle of this malleus moves inward, and stapes bone, this time it does not move there to the oval window, it moves outward like this. Handle of malleus moving inward, stapes bone moving outward like this, and because of this, the ossicular chain goes into rigidity. Look, the stapes bone is not moving towards the oval window, it's moving outward like this, and it creates a rigidity in the ossicular chain, and it dampens the pressure in that sound wave, so that damage to the inner ear and cochlea will be prevented. So these are the middle ear mechanisms. Let's see the inner ear mechanisms now. Inner ear has cochlea. Well, there is labyrinth in the inner ear. On one hand, there is vestibular apparatus, on one side, and then associated with is the cochlea. So cochlea and vestibular apparatus makes the labyrinth in the inner ear. We are now talking about organ of hearing. So cochlea, let us see the parts of the cochlea and mechanism of hearing. The base of the cochlea is called as limbus. Then the bony center of the cochlea is called as modiolus, and the apex of the cochlea is called as helicotrema. Helicotrema. There are these three chambers: scala vestibuli, scala media, and scala tympani. Scala vestibuli and scala media are separated by Reisner's membrane, and scala media and scala tympani are separated by basilar membrane. So Reisner's membrane and basilar membrane. Vestibuli and tympani, they become one chamber at the apex of the cochlea. That's called as helicotrema. This entire chamber, scala vestibuli and tympani, it's filled with a fluid called as perilymph, and due to its connection with the subarachnoid space, it has a composition which is very similar to the CSF. Scala media is filled with another fluid called as endolymph. We have already discussed much about endolymph. It has got rich potassium concentration, and therefore, in spite of the fact that it is ECF, extracellular fluid, it more resembles ICF. Now, on the basilar membrane, there are basilar fibers. So what is going to happen is, when the sound waves come through the middle ear and through the oval window, they will fall on the basilar membrane. Reisner's membrane is almost non-existent, as if it is non-existent. Sound wave will cross it and fall on the basilar membrane. Basilar membrane vibrates, therefore these basilar fibers will also vibrate. And now we will see how the pressure in the sound wave will be converted into electrical action potentials. Transduction of sound. So transduction of sound is caused by the organ of Corti, which is present on these basilar fibers. Before we discuss the organ of Corti, let me just explain this, that the basilar fibers near the base of the cochlea, they are short and thick, and as you move towards the apex, the fibers become long and thin. So the high frequency waves, when they enter the inner ear, they will excite the basilar membrane near the base. The basilar fibers near the base will vibrate maximally because they are short and thick. So high frequency waves will stimulate those fibers maximally. Whereas fibers at the apex are long and thin. So low frequency waves will cause maximum vibrations of the fibers near the apex because there the fibers are long and thin. So low frequency sounds will vibrate those fibers maximally. On those basilar fibers, there is organ of Corti, which is the organ for hearing, which causes transduction of sound. Pressure wave will be converted into electrical potentials by the organ of Corti. So here is a bony center of the cochlea, the modulus, and the basilar fiber arising from it. On the basilar fiber, there is organ of Corti, which appears like this. There are rods of Corti. There is reticular lamina, and then there is a sheet of tissue called as tectorial membrane. A sheet of tissue called as tectorial membrane. There are hair cells in the organ of Corti. There is a single row of inner hair cells, and there are 3 to 4 rows of outer hair cells toward the open end of the basilar fiber. 3 to 4 rows. In each row there are about 3000 hair cells. We have shown a single row in the form of single hair cell, but there are 3000 inner hair cells and 9 to 12,000 outer hair cells, because in each row there are 3000 hair cells. It is the inner hair cell which is concerned with the hearing. Outer hair cells are going to create the receptor potentials, which are called as cochlear microphonics. Receptor potentials are called as cochlear microphonics, which are created by the outer hair cells. Sides and bases of the hair cells synapse with the 8th cranial nerve fiber. So, how the transduction of sound will occur, means how it will be converted into electrical potentials, is this. When the basilar membrane vibrates due to the sound energy, the pressure in the sound will cause vibration of the basilar fiber, and as the basilar fiber vibrates, there will be shearing force generated between the reticular lamina and tectorial membrane. Reticular lamina and this sheet of tissue called as the tectorial membrane. There is a shearing force created, and that shearing force will cause bending of the stereocilia. These hair cells have these hair called as stereocilia. So shearing force created due to the vibration of the hair cell will cause bending of the stereocilia, and there are potassium channels at the top of the hair cell. Bending of the stereocilia will open the potassium channels. Potassium will enter and the hair cell will depolarize. Ionic current has been established. So sound wave has been converted into electrical energy, electrical potential. Finally, as hair cell depolarized, it will open the calcium channels. Calcium will come in, and glutamate, the neurotransmitter, will released by exocytosis. It will excite the auditory nerve fiber. So this is how sound wave, or that energy, the pressure in the sound wave, is converted into electrical potential. And finally, 8th cranial nerve fiber, the cochlear nerve or auditory nerve, will take the impulses to the auditory cortex in the temporal lobe. That is when we will hear the sound. So that's the mechanism of hearing. Let us also discuss the equilibrium, vestibular apparatus and the process of equilibrium. There was one question like this. A patient is on a stretcher and the stretcher is in an elevator starts going up. Which part of his vestibular apparatus will be stimulated maximally? And the options are macula of the utricle, macula of the saccule, vertical semicircular canal, horizontal semicircular canal. Let's try to understand the answer now. With the process of equilibrium, the vestibular apparatus. Basically, the labyrinth, petrous part of the temporal bone has bony labyrinth, and inside the bony labyrinth there is membranous labyrinth. The membranous labyrinth has two parts. One is the cochlea, which we saw, the organ for hearing, and the other part is vestibular apparatus. So the vestibular apparatus has these structures: utricle and saccule, and then the three semicircular canals. So the organs for equilibrium are utricle and saccule and three semicircular canals. So this is the cochlea, that's the vestibular apparatus. Now, utricle and saccule, let's look at these parts one by one. Utricle and saccule, they play the role in equilibrium when the head is stationary or a linear acceleration of the head. Whereas semicircular canals, they are important in equilibrium. They maintain the balance of the body when there is angular rotation of the head. One more difference between the two is that semicircular canals have a predictive function, which the utricle and saccule do not have. So if you are running and there is going to be a sharp turn, it can be predicted 1 or 2 seconds in advance so that the appropriate balance of the body can be maintained. This is how we can maintain the balance even when there are sharp turns or sharp changes in our directions. It is predicted 1 or 2 seconds in advance by the semicircular canals. Utricle and saccule involved in the linear acceleration, means maintaining the balance of the body when there is linear acceleration. Inside them, there is the organ for equilibrium called as macula. Macula inside the utricle is horizontal, like this. This is the macula inside the utricle. It is horizontal. Macula inside the saccule is vertical. Inside the maculae, there are receptors for equilibrium called as hair cells. So that's the orientation of these maculae and therefore orientation of the hair cells. So first of all, try to understand this. This is a person, let us say it is the person seated in a train and the train suddenly starts. This is the horizontal linear acceleration. It will be detected by the macula inside the utricle. So remember, macula inside the utricle will detect horizontal linear acceleration. how it will maintain the balance, how it will detect the linear acceleration is that when the person is suddenly thrust forward, when the train begins. Now, first of all, just understand what all things are also present in this macula. At the top of the macula, there are calcium carbonate crystals, which are called as otoconia, or also called as tatoconia, and therefore these are called as otolith organs. Ear dust or otolith organ. These are crystals, stony hard crystals. They have very high specific gravity as compared to the hair cells. So imagine, and also sides and bases of the hair cells, they synapse with the 8th cranial nerve fiber, vestibular nerve fibers. So when there is horizontal linear acceleration, head is thrust forward, macula inside the utricle, it will move forward. But initially the calcium carbonate crystals, is otoconia, will not move. They have a high specific gravity, and therefore since they have not moved initially, there will be bending of the stereocilia like this. Stereocilia will bend. They were studied in those otoconia at the top of the maculae, at the top of this macula. So when the head is thrust forward, hair cells will move immediately as the head is thrust forward, but those otoconia will not move immediately, and therefore it is a relative opposite movement. Means as if the otoconia are going back. Actually hair cell is moving forward and these are still, the otoconia are still, because they have high specific gravity. The crystals are still. But this is taken as a relative opposite movement, means as if otoconia are going back, and therefore there will be bending of the stereocilia. This will excite the hair cell by entry of calcium, and finally, as we have already discussed, glutamate will be released and there will be stimulation of the vestibular nerve fibers. Signals will be sent to the vestibular nuclei in the brain stem, and then vestibulospinal tract comes down on the spinal cord. It controls the anti-gravity muscles of the body. We will contract the anti-gravity muscles, and as the train has been thrust forward, we will contract the muscles so as to maintain the balance in the opposite manner. So that's horizontal linear acceleration. Vertical linear acceleration, this is macula inside the saccule, plays the role in equilibrium with vertical linear acceleration. Imagine this person, this is the head of the person, and this is the macula inside the saccule. So when suddenly the lift starts going up, the elevator starts going up suddenly, what will happen? The same thing. Hair cells will start moving upward vertically, but otoconia will not move, and therefore there will be bending of stereocilia in the opposite direction. The potassium channels will be opened at the top of the hair cell. Potassium will enter, hair cell will depolarize, and that will finally excite the vestibular nerve fibers, and it will send signals to the vestibular nuclei in the brain stem. Vestibulospinal tract will control our anti-gravity muscles, and we will maintain the balance. So in summary, horizontal linear acceleration, the organ for equilibrium is macula inside the utricle. Vertical linear acceleration, the organ for equilibrium is macula inside the saccule. Now imagine that the patient was on a stretcher. This is a man who is vertical. When the man becomes horizontal, then macula inside the saccule will become horizontal, and macula of the utricle will become vertical. It will go in this position when the man goes in horizontal position. So macula of the utricle will now become vertical when the man lies down. This is a man standing. When man lies down, this will become horizontal. the macula of the saccule, and macula of the utricle will become vertical like this, in this position. And then when the lift goes up, macula of the utricle will be stimulated maximally. So answer to that MCQ was macula of the utricle will be stimulated when the patient is in on the stretcher and it starts going up. Finally, angular rotation is detected by semicircular canals. There are three semicircular canals: anterior vertical, posterior vertical, and horizontal. Anterior vertical, posterior vertical, and horizontal. They represent three planes in the space. They are all at right angles to each other, and they are representing three planes in the space: anterior vertical, posterior vertical, and horizontal. They are all at right angles to each other. If you look at the semicircular canals, one end of each canal is dilated. This dilated end is called as ampulla. Inside the ampulla, there is a crest-like elevation called as crista ampullaris, and within the crista ampullaris, there are hair cells. Now, note this point that in the utricle and saccule, all the hair cells were lined up in the same direction. They were lined up in the same direction. Whereas in the semicircular canals, they are oriented in all the possible directions. And that is why semicircular canals play an important role in equilibrium when there is angular rotation of the head. Sides and bases of the hair cells, they synapse with 8th cranial nerve fibers, vestibular nerve fibers. And top of the crista, there is the organ called as cupula. Cupula. So, how is it that we maintain the balance during angular rotation of the head? Also, how is it that there is nystagmus during the rotation? Let's try to understand this. When we start rotating the head, let's say in this direction, in a particular angle, entire semicircular canal will start moving, but endolymph within the semicircular canal does not move initially. It does not move with the same speed. It does not move initially. So because endolymph is not moving, but entire semicircular canal begins to move in the direction of rotation, therefore consider this. This is a relative opposite movement. Again, endolymph is stationary, but canal is moving forward, and therefore there will be bending of the cupula, because endolymph will now push the cupula like this. Semicircular canal going ahead, endolymph stationary. It will push the cupula backward. This bending of the cupula will cause the depolarization of the hair cells by movement of the stereocilia in one direction and opening of potassium channels. Bending of the cupula will cause bending of the hair cells, bending of the stereocilia and opening of potassium channels. As we have already described, these are the stereocilia. When they bend, the potassium channels at the base of the stereocilia will open, potassium enters, hair cell is depolarized. So, and as the hair cell is depolarized and finally it excites the vestibular nerve fiber, the signals are sent to the vestibular nuclei and to the cerebellum. Vestibulospinal tract, from vestibular nucleus to the spinal cord, will control the anti-gravity muscles and control the posture. That is how we can control the posture when there is angular rotation of the head. Apart from this, the vestibular signals are also responsible for vestibulo-ocular reflex, the rotatory nystagmus, the vestibulo-ocular reflex. As we rotate the head on to the left, let's consider that rotating the head to the left continuously will cause eyes to slowly deviate to the right. When they go to the extreme of the orbit, they quickly return to the center. If the head is still rotating, eyes will slowly deviate to the right, that is in the opposite direction, then quickly return to the center. This will create oscillatory movements of the eyes, create the oscillatory movements called as nystagmus, the rotatory nystagmus. It will continue to occur as long as the cupula is bent. Now, what will happen eventually is that endolymph will begin to move. As you are rotating the head in one direction, initially the endolymph was not moving, and that bent the cupula in the opposite direction. That is why the eyes were slowly rotating to the opposite direction and then quickly returning to the center. In 10-15 seconds, endolymph will catch up and it will start moving in the same direction with the same speed as the head is going. Cupula will come back to its original position and the nystagmus will stop. Those rotatory movements of the eyes, oscillatory movements of the eyes will stop. When the head stops rotating, now the opposite will occur. Now the semicircular canal has stopped, but endolymph continues to move for some time in that same direction. And now therefore cupula will bend in the opposite direction. Now initially when head started rotating, the cupula was bending like this because head was going like this. Now endolymph is continuing to move even though head has stopped moving. Therefore now the cupula bends like this, and that will again generate the signals in the vestibular nerve, and this time it will be called as post-rotatory nystagmus, the jerky oscillatory movements of the eyes. If your head was rotating to the left, then the eyes slowly deviated to the right and then quickly returned to the center. By the way, this quick movement, fast movement defines the direction of nystagmus. If it is going to the left, it is called as left nystagmus. Then when the head stops rotating, there will be a post-rotatory nystagmus because endolymph continues to move and it bends the cupula now in the opposite. Therefore now the slow movement will be to the left and fast movement will be to the right. If your head was moving to the left, then when it stops, now the slow movement will be to the left and rapid movement will be to the right. That's called as post-rotatory nystagmus. It's in the opposite direction to that of the nystagmus that happened at the start of the movement. There is also something called as spontaneous nystagmus of labyrinthectomy. Because of the impulses going on by our head detection, there are spontaneously the impulses are going on in the 8th cranial nerves on both sides. Because of the impulses in the left labyrinth and left 8th cranial nerve, the eyes slowly rotate to the right, slowly deviate to the right, and because of the impulses in the right, the eyes slowly deviate to the left. Now these signals cancel out each other and we have a parallel gaze normally under normal circumstances. Now imagine there is unilateral labyrinthectomy, that is left side labyrinth has been removed. Then what will happen? There are no signals in the left vestibular nerve. That means there will be signal in the right vestibular nerve because of which the eyes will slowly deviate to the left and then quickly return to the center. Slowly deviate to the left and quickly return to the center. This will create the spontaneous nystagmus of labyrinthectomy, unilateral labyrinthectomy. And finally, one more condition that produces nystagmus oscillatory movement is the temperature, and its mnemonic is cows, as you must be aware. Cold, opposite, warm, same. If you pour cold or warm water into the external ear, because of the difference in temperature between this water and the endolymph, the convection currents are generated in the endolymph, and that those convection currents, they excite the hair cells in the vestibular apparatus, and they cause a spontaneous oscillatory movement of the eyes called as spontaneous nystagmus. And it is written as this: if it's a cold saline, then it is opposite sided nystagmus. Warm saline, same sided nystagmus. And remember, side of the nystagmus is defined by the fast component of the nystagmus, as I have already mentioned. For instance, if you are rotating your head to the left, eyes slowly go to the right. Why do they have to go to the right slowly? First of all, why it happens is because it has to have a good visual fixation. Imagine if your head is rotating and eyes also move along with the head. What will happen? There will be quick passing of visual scenes across the retina. That's not what we want, and therefore when the head is rotating to the left, eyes will slowly go to the right with a one visual fixation point. When they reach the extreme of the orbit, then they quickly return to the center of the orbit to have the new visual fixation. If your head is still rotating and there is still the movement of the endolymph, then again same thing will happen. Eyes rotate in the opposite direction slowly and quickly return to the center. The quick component defines the side of the nystagmus, remember. So if you pour cold water in the ear, there will be opposite sided nystagmus. Means fast component will be on the opposite side. Slow movement will be on the same side. Means if you pour cold water in the right ear, eyes will slowly go to the right and then quickly return to the left. That's cold, opposite. Warm, same. Warm water will produce same sided nystagmus.